And good afternoon, everyone. I'm Dr. Kim Allen Williams, Chair of Medicine here at University of Louisville, and it's my pleasure this afternoon to introduce Research Grand Rounds. And it's going to be given to us by Timothy Edward O'Toole. He is our PhD in our Envirome Institute, the Christina Lee Brown Envirome Institute, I should say, in a division of the Department of Medicine. He comes to us after being a Wolverine for a while. Did chemistry, cell biology at the University of Michigan, then a PhD in biological chemistry there. Did his postdoctoral fellowship in immunology at Scripps Clinic in La Jolla. And he worked his way up the academic ranks until he actually came to be an assistant professor. My goodness, that's 19 years ago. How about that? Here at University of Louisville. And he has been an associate professor since 2019 in the Envirome Institute. And he's going to speak to us today about microplastics exposure and cardiovascular disease. New England Journal of Medicine, March 7th, 2024.
All right, I'm going to talk about that.
I'm so glad you're doing this.
Thank you for the introduction. And I'm sure on behalf of everyone, we'd like to thank Dr. Williams for our lunch. But as we use our utensils and drink our favorite beverage, I'm going to hope to convey that this might not always be the best thing for us. Yes. So I think as we're all aware, there's a huge usage in modern society of plastic products. A lot of this comes from food packaging, commercial products, consumer products, but also when we think about what we do in the lab and in the clinic, there's a lot of single-use plastics. So although there are programs to recycle plastics, a lot of these used plastics end up in landfills and the water systems. And I've put here some numbers. Over 360 million tons of plastics are produced annually, and a large amount of this, 4.9 billion metric tons, end up in aquatic systems or the landfills. Under these storage conditions, there's a gradual degradation of these large polymers into smaller microplastics or nanoplastics. This occurs both through physical chemical processes, the abrasion, some oxidation, some exposure to UV light. And under these processes, these larger polymers are broken down into what's known as the secondary microplastics. In addition to these secondary microplastics, there's also contamination with primary microplastics. And this refers to plastic particles of the size range, which are synthesized directly in the size range for commercial purposes. And in these conditions, in the aquatic and terrestrial ecosystems, it's quite easy for these particles to enter into the water supplies and the food chains. So indeed, there's been several studies suggesting that microplastics have been found in human tissues, and there are three general ways where humans are exposed. The first is through ingestion, and some studies suggest that humans can consume as much as 5 grams per week, which is a weight of a typical credit card. Because they are lightweight and of low density, microplastics can also become airborne. Once they're airborne, we can inhale these. What comes up must come down, so these particles also can fall out onto vegetation. And indeed, a couple of years ago, there was a study in some western parks in the western part of the United States, which developed the concept of plastic rain as a new form of acid rain. And finally, although maybe somewhat less abundant, there's also some exposures through absorption. As I mentioned, several studies now have identified microplastics in multiple human tissues. Interestingly, a recent study published just a month ago from a group of New Mexico and Nature Medicine suggested that the highest levels of microplastics were found in the brain. This was done on some tissues collected from autopsy. And interestingly, it has been found in the study, they also documented that the highest microplastics concentrations were found in those who presented with a dementia diagnosis. Our interests have been in the effect of microplastics on cardiovascular disease. And this interest was spurred by 3 papers that came out in the early part of last year, which I'll go through now. The first paper, as Dr. Williams alluded to, was a New England Journal of Medicine paper in March of a year ago. This was a study from an Italian group where they collected 304 endarterectomy samples. And in their analysis, they detected that 60% of these samples had detectable levels of microplastics. And of the microplastics, the highest levels were polyethylene and polyvinyl chloride. They composed by weight, they found that up to 2% of the plaque by weight was composed of these microplastics, a pretty high level. And this was a prospective study, so in some follow-up, they found that those with detectable levels of MP were at higher risk of an endpoint event. A second study, which came out about the same time, looked at levels in coronary arteries and carotid arteries in those with plaques and without plaques. And in this study, they found that in all samples, they did find detectable levels of microplastics, but of somewhat less abundance, 0.01% by weight compared to this 2% in the New England Journal of Medicine paper. In this study, the most abundant plastic types were PET, polyamide, polyethylene, polyvinyl chloride. And again, they found that higher levels were found in tissues that were obtained from plaques than those without plaques. And a final study later that last year, blood was obtained from 19 controls, as well as 82 subjects who had acute coronary syndrome. Again, quantification of microplastic types. They predominantly found that polyethylene and polyvinyl chloride were the most abundant. And they found that levels in those with risk of coronary artery disease was higher than levels in those who had lower risk. And also the levels in those with acute MI were greater than those in stable angina. So we were interested, given these studies, and we were interested in what type of functional effects microplastic consumption might initiate. So some of the earlier studies about the physiological consequences of microplastic exposure were done in aquatic animals or in rodents. And some of the most abundant or the most common outcomes were that ingestion promotes inflammation, increased oxidative stress, it induced changes in the gut microbiome, and it altered cellular bioenergetics. So since these physiological outcomes are known to promote CVD risk, we simply asked, does indeed consumption promote CVD risk? And furthermore, does ingestion of microplastics cause overt disease? So to do these studies, we use an animal model. We use the C57 black 6 mice. We gave them a normal water or water containing polystyrene microplastics. In this initial study, we used two different sizes of beads, 5 micron and 0.5 micron, and two different doses of each bead size, 0.1 microgram per mil and 1 microgram per mil. And these values, these doses and sizes, we can make back of the envelope calculations and kind of estimate how many plastic particles these sizes and doses constitute. And based upon some earlier human studies upon the exposure levels, we can make these calculations and kind of simulate what's going on in a human in this mouse model. So we gave these water supplies to the mice for 12 weeks. During this time period, we recorded several measures at different time points, the glucose levels, the weight, metabolic activity. In one study, we also performed an echocardiogram. And then at termination, after 12 weeks, we can collect blood in several different tissues and do several measurements of metabolic activity and cardiovascular health. So first, just some general measures of what's going on during the feeding time course. We housed mice in a metabolic chamber for 24 hours. And what we saw, although there were some slight differences in food intake and water intake in the two groups, there was really no difference in the respiration rates or in the metabolic activity of mice exposed to either the normal water or the polystyrene containing water. So over the next few slides, I'll talk about some of our endpoint measures and some of the significant findings. First of all, we found that there was a disruption of glucose homeostasis. On the far left, we measured fasting plasma glucose. And although I think this particular graph was done at termination, we see these changes as early as 3 weeks of feeding. So we observed that several of our treatments promoted a fasting plasma glucose. And it seemed that the high dose, the 1 microgram per ml dose of either bead size, promoted the greatest increase in fasting plasma glucose. At termination, we also saw, again, in the mice drinking the smallest size particle at the high dose, we saw an increase in fasting plasma insulin. As you might expect, the HOMA-IR scores were also the highest in this same exposure group. A second outcome we observed was an increase in adiposity. So again, this is the graph on the left is a measure at termination, but we saw the same changes, are smaller in magnitude, but the same kind of trend after 3 weeks of exposure. And that is at the highest dose, the 1 microgram per ml dose of either bead size, we saw an increase in body weight. When we did an analysis of these mice using the DEXA scan, we saw that this increase in weight was due an increase to adiposity. And again, we see that several groups show an increase in adipose tissue weight. Because of this indication of adipose dysfunction, we collected the adipose tissue at termination and did a flow cytometry assay to look at the presence of immune cells in the adipose tissue. Using an F480, a marker of total macrophages, we saw that there was a significant increase in mice consuming the 0.5 micron beads and a trending increase in those mice consuming the larger size bead. We next asked whether this increase in macrophages was specifically due to the M1 pro-inflammatory phenotype. And although there was not significance, there was a trending p-value of 0.07 increase of the M1 pro-inflammatory macrophages in the adipose tissue. Although we see these changes in weight, the increase in glucose and insulin, the suggestions of adipose dysfunction, when we measured adipokines in plasma or measured certain indices of endothelial activation, the circulating levels of ICAM, P-selectin, or E-selectin, we did not see any change between our treatment groups. Nor did we see any major change in lipid levels, although in one group we did see an increase in HDL levels. Because of this indication of adipose dysfunction, we wanted to look at gene expression changes, see if there was anything upregulated or downregulated that might indicate an increase in adipogenesis. So we did this analysis in the perivascular adipose tissue, or PVAT. These are results from a targeted microarray analysis. And what we observed was that there were 14 genes that were decreased by greater than 1.5-fold. Those in red are those that are involved in the inhibition of adipogenesis. So if they are down, that would mean that adipogenesis is promoted. In addition, we saw that 5 genes were increased by greater than 1.5-fold. The most prominent of these is DKK1, which is an inhibitor of Wnt signaling. And when Wnt signaling is inhibited, there is a promotion of pre-adipocyte differentiation into mature adipocytes. And finally, we saw that there were minor changes, but perhaps a trending increase in certain inflammatory genes, IL-6 and MCP1, in the adipose tissue. The next change I wanted to mention were changes in the microbiome. So we collected feces throughout the feeding time course and measured by 16S sequencing done by facilities here at UofL. We looked at the microbiome content. There were 2 significant observations. First of all, and that in mice consuming the polystyrene water, there's a decrease in beta diversity. Secondly, when we looked at the genus abundance, we saw that there were changes that specifically that there's an increase in the polystyrene fed mice. There's an increase in the firmicutes over bacteroidetes. So these 2 findings, the changes in diversity and the relative abundance of the genus are consistent with what has been shown in the literature. That is, the consumption of a high-fat Western diet is associated with decreased bacterial diversity. Similarly, in both obese humans and mice, we see these changes in the relative abundance of certain bacterial genus. In collaboration with Matt Cave, we also wanted to look at effects on hepatic biology in our polystyrene-fed mice. So interestingly, although we see these changes in adiposity and glucose handling, there was no outward change in liver histology, although there was a slight increase in cholesterol levels in both in mice consuming either size bead. We also measured bile acids, and this is just a large table showing the bile acids that were measured and their levels. There were changes in distinct bile acid levels. I just wanted to denote two. One in red, which shows that there's an increase of HDCA in mice consuming the 5 micron bead size, while there's a decrease in the 0.5 micron bead size. And another interesting finding was that there seemed to be differences in short chain or in bile acid levels depending upon where they're measured. For example, in blue, in the cecum, we see relatively no change, whereas it's decreased in the liver. So we are interested in trying to decipher what these changes in bile acids mean with respect to the physiology of the whole animal. And finally, I wanted to point out that we did some RNA-seq on the liver. We saw that several pathways were upregulated in the 0.5 micron in the left-hand side of the figure in red, as well as, and there was also some changes in samples obtained from the 5 micron, a few or less. But what was notable was that in both bead sizes, there was activation of FXR, RXR, LXR activation. And again, we are interested in trying to pursue this and with respect to what it means to the physiology of the whole animal. And the final outcome I'd like to measure was in one study, we did a echocardiogram. What we observed was that there were increases in the wall thickness, corresponding decreases in the internal diameter, with changes in the stroke volume. Interestingly, these changes were observed almost exclusively in mice drinking the large size microplastic bead compared to the smaller size bead. So this suggests that there might be some size dependent outcomes. If you remember before, most of my metabolic changes we observed in the 0.5 micron size beads. Here, it seems to be the opposite. So it does seem like drinking polystyrene beads promotes some of the risk factors for cardiovascular disease. So now we wanted to ask if it would in fact increase frank disease, focusing on atherosclerosis. So to do these studies, we used the atherosclerosis-prone mouse strain, the ApoE KO mice. Again, we fed them or supplied them with either normal water or water containing polystyrene. In these exposures, we only used one bead size and one dose, that size and dose which gave the greatest metabolic defects. We've placed these mice on normal chow for 20 weeks. Again, during this time course, we recorded several measures, and at termination, we looked at lesion size, characterized lesions, other measures of cytokines, complete blood cell counting, as well as these plasma biomarkers. So here are some of the significant outcomes. The first and most surprising thing was we saw that consumption of the polystyrene beads promoted lesion formation. On the left-hand side are just two oil red stain images from a mouse drinking normal water or polystyrene containing water. And when we quantified the lesion area, we see that there's a significant increase in the lesion area in the polystyrene fed mice. Using aorta from these mice, we also generated RNA, did a crude crude RNA prep from the ARTA, which would be of multiple cell types, but we nevertheless did an RT-PCR for other markers characteristic of lesion formation. And what we saw that there was an increase in smooth muscle actin, suggesting a smooth muscle proliferation. There was a small increase, but in CD68, a macrophage marker, and an increase in MMP9, suggesting that these lesions may be somewhat unstable. As with our earlier liver samples, we collected the aortas, did some RNA-seq on the aortas. Here is on the right-hand side of the slide is just a large table of the pathways that are changed upon polystyrene consumption. And what we saw was what you might expect, that there was an increase in lymphocyte proliferation, lymphocyte activation. So some of the typical indices of a pro-inflammatory phenotype. We also measured plasma cytokines and plasma lipids. However, we saw really no change in any of these measured parameters. So we see that polystyrene consumption promotes both CVD risk factors as well as overt CVD. And next, we turned our attention into what might be the mechanism for these outcomes. And in looking at our data, we've come to focus on really three different mechanisms. The first would be that consumption of these plastic particles promotes an altered gut microbiome. And this is just data you've seen before from that shows that there's decreased diversity as well as changes in the genus. So what might be the mechanism whereby an altered microbiome can promote CVD and atherosclerosis? One is that change the microbiome can affect central appetite. It can affect fat storage. It can promote a chronic inflammatory state. And finally, that it might alter the levels of short-chain fatty acids. These short-chain fatty acids are microbial metabolites. They're produced by the fermentation of dietary fiber. And interestingly, it has been found through several studies that these can protect from or are associated with obesity and atherosclerosis. So we collected from the plasma that was collected, we did an analysis of short-chain fatty acids. And what we observed that there was a significant decrease in propionic acid. Propionic acid is important because most studies suggest that it's protective. That is, it can reduce intestinal cholesterol absorption. It can stimulate the expression of anti-inflammatory cytokines, and specifically IL-10. There were no change in other short-chain fatty acids, such as butyric acid, nor was there a change in TMAO, another bacterial metabolite that is associated with an atherosclerotic phenotype. So in thinking about how to address this mechanism in future studies, one way to get more insight as to whether the microbiome is important would be to do fecal transplant experiments. That is, take the feces of exposed animals, transplant that into a naive animal, and see if these naive animals also develop these phenotypes. A second way to address this mechanism would be to probiotics supplementation, specifically with certain subclasses of lactobacillus substrain, which has been specifically reported to protect from some of these changes in atherosclerosis and hyperglycemia. And finally, if we could increase levels of propionic acid, we might be able to reverse these effects induced by polystyrene. And one way, there are several ways to increase propionic acid. One way would be dietary supplementation with xylitol. A second potential mechanism for what we're observing is that it might, consumption of microplastics might disrupt the intestinal barrier. So under normal conditions, there's an intact intestinal epithelium. It prevents the leakage of deleterious bacteria or metabolites into circulation. And these tight junctions are maintained by a series of several proteins. However, under conditions where this barrier is disrupted, you have a leaky gut, what's known as a leaky gut syndrome, where these deleterious bacteria and metabolites leak into the circulation. When this occurs, there's a whole series of pathological outcomes can result, including hepatic biology, changes in adipose biology, insulin resistance, and chronic inflammation. So to look at whether the intestinal barrier was disrupted in our animals, we used this common FITC dextran assay. We gavage the animals with FITC dextran. After 4 hours, we collect blood and then simply do a measurement of the fluorescence in the collected blood. And when we did this with our polystyrene treated animals, although there was somewhat of a wide distribution of data points, there was a significant increase in FITC dextran in the polystyrene fed animals, suggests there is, in fact, some disruption of the intestinal barrier. Another bit of evidence for disruption of the intestinal barrier came from an RNA-seq analysis. So we collected aorta from these animals, did an RNA-seq, and one of the most downregulated genes, in fact, the most downregulated gene, was vasoactive intestinal peptide, or VIP. So VIP can be expressed in both the gut and in the vascular tissue. In the gut, it regulates the expression of these intestinal tight junction proteins, maintaining the intestinal epithelial barrier. And it also promotes microbial homeostasis. In the vasculature, VIP has been shown to decrease the expression of pro-inflammatory cytokines, as well as suppress leukocyte activation and migration. So VIP appears to play very protective roles, and in our system with polystyrene, it seems to be downregulated in expression. And a final mechanism we are looking at is the possibility that polystyrene could impact endocannabinoid signaling. The endocannabinoids are largely expressed in the brain, where they are involved in several functions, such as memory, learning, appetite, emotional processing, sleep, and pain control. However, the elements of the endocannabinoid signaling are also expressed in other cell types and tissues, such as adipose, smooth muscle cells, and endothelial cells. And in these cell types, it has the properties of regulating inflammatory responses and regulating metabolism. So there are two endogenous endocannabinoids, anandamide, or AEA, and 2-arachidinoglycerol, or 2-AG. There's another compound, PAE, which has endocannabinoid-like properties. These endocannabinoids are synthesized on demand from membrane fatty acids. AEE is synthesized through the enzyme NAPE. It is degraded through fatty acid amino hydrolase, or FAAH. Similarly, 2-AG is synthesized by DAGL and degraded by MAGL. There are two known endocannabinoid receptors, two G protein coupled receptors, CB1 and CB2, and there's a third endocannabinoid receptor-like molecule, GPR55, which binds PEA. These receptors are somewhat promiscuous, and then they can bind multiple endocannabinoids, although it appears that AA prefers to bind to CB1 and 2-AG prefers to bind to CB2. The signaling properties of these endocannabinoid receptors are different. When signaling proceeds through CB1, that is thought to be pro-inflammatory, inducing an atherogenic-like phenotype. And this has mostly been shown through the use of knockout mice or use of specific inhibitors of CB1. In contrast, signaling through CB2 is anti-inflammatory and seems to be protective. So our interest in the endocannabinoid signaling was again spurred by our RNA-seq results. Two other genes which were highly downregulated were CNR2, the gene that encodes CB2, and GPR55. In addition, we did some targeted RT-PCR looking at the metabolic enzymes for these endocannabinoids. While there was an increase in these synthetic enzyme DAGL, there's also a compensatory increase in MAGL, so changes in 2-AG would be minimal. However, we saw an increase in the biosynthetic enzyme NAPE and a decrease in the catabolic enzyme FAH, suggesting that AEA would be increased. So given that anandamide, or AA, was increased together with a downregulation of CB2 and GPR55, this would suggest that endocannabinoid signaling would proceed primarily through CB1, which is pro-atherogenic. And interestingly, there's some studies suggesting that CB1 signaling is also promotes cardiac hypertrophy. So how does endocannabinoid signaling, and specifically through CB1, promote atherogenesis? One possibility is that CB1 signaling promotes inflammation. It stimulates the production of inflammatory cytokines by leukocytes. A second possibility is that it promotes endothelial dysfunction and the expression of adhesion molecules. A third possibility is that endocannabinoids promote smooth muscle cell proliferation in atherosclerotic lesions. And finally, endocannabinoids can promote cholesterol uptake and accumulation, especially in the macrophages in lesions. So the idea that environmental exposures can affect endocannabinoid signaling is not that far-fetched. There's been previous reports that bisphenols, cigarette smoke, arsenic, lead, phthalates, and organophosphates all promote or all influence the expression of the endocannabinoid receptors and or the expression or activity of the endocannabinoid metabolizing enzymes. So when thinking about how to address this mechanism, there are some possibilities that we are considering for future experiments. One is the use of these CB1 knockout mice, which would be predicted to be resistant to the pro-atherogenic effects of polystyrene. A second possibility is the use of CB1 antagonists, and there are several that have been used. And another way would be to increase expression of the catabolic enzyme for AEA, increase expression of FAH, either by leptins or by steroids. So I've talked about these mechanisms as if they operated separately and independently. That is not always the case. For instance, endocannabinoid signaling, especially with CB2, can increase expression of VIP. It also can stabilize the microbiome. So trying to identify a true mechanism of polystyrene-induced pathology may be somewhat difficult, as polystyrene can affect many of these pathways as I've seen. But we're going to try and do what we can to try and identify what's going on in our polystyrene exposures. Okay, so what about other plastics? I've talked to you about polystyrene. That is an abundant plastic found in the environment. However, it is not the most abundant. As I mentioned in the earlier studies from collected cardiac and tissue and from blood tissue, the most abundant plastic types seem to be polyethylene and polyvinyl chloride. And these are just some data from those two papers. So what about these two plastics? So again, we address this using a similar experimental protocol. This should be ApoE mice, not C57, but we gave ApoE mice normal water or that containing polyethylene and polyvinyl chloride. In this study, we tried to mimic the size and dose as was found in the human studies. So we used these bead sizes and doses. And again, we were somewhat in a hurry to get this, get data from this experiment. So rather than putting mice on a normal diet, we put them on a Western diet for 6 weeks. Again, during this feeding time course, we made several repetitive measures. And at termination, again, has several parameters which we wished to measure. So here are some of the outcomes. As with polystyrene, we saw that feeding with PE plus PVC induced fasting plasma glucose levels as well as fasting plasma insulin levels. So again, a metabolic phenotype. When we looked at the lesion size, collected the heart valves at termination, stained with red O. Again, we see, similar to polystyrene, we see that there was increased lesion size in the mice consuming the polyethylene and polyvinyl chloride microplastics. Again, from the aorta, we collected aorta, isolated RNA, looked at elements of endocannabinoid signaling. Consistently with the polystyrene results, we saw an increase of the anabolic enzyme NAPE and a decreased expression of the catabolic enzyme FAH, again, suggesting that endocannabinoid levels were increased. Interestingly, this exposure, we also saw an increase of CNR1, the gene expressing CB1. So again, we have a suggestion here that endocannabinoid signaling is impacted by the consumption of PE plus PVC. However, we also saw some differences in this exposure. In contrast to polystyrene, where we saw a decrease of propionic acid, in this exposure, there was no change in propionic acid levels or in changes of other short-chain fatty acids. As mentioned, we also did a CBC. In all of our polystyrene exposures, we saw no changes in complete blood cell counts or in the red cell parameters. However, in the PE plus PVC exposure, we did see a significant decrease in red cell levels, as well as hemoglobin content and hematocrit. So this suggested that there might be microplastic type specific outcomes. So our consumption of microplastics is inevitable. Might there be something we can do to overcome these effects? Specifically, might there be some dietary interventions that we could enact that would limit oxidative stress, inflammation, and altered microbiome, some of the outcomes that microplastics seem to induce. So in searching the literature, one of the compounds that seemed favorable to us was delphinidin. This is a flavonoid anthocyanin that is found in many fruits and vegetables, and gives colors. It's a purple, deep red color. It gives color to some of these common food groups. Interestingly, delphinidin has antioxidant, anti-inflammatory, and anti-angiogenic properties. In searching the literature, it had several of the properties which we were interested in. Specifically, when mice on a high-fat diet were given delphinidin, they exhibited several metabolic changes. Specifically, a decrease in glucose levels, decreased lipid accumulation, reduced weight gain, and inhibition of adipogenesis. Delphinidin also had some anti-inflammatory effects, reducing the expression of anti-inflammatory cytokines and decreasing leukocyte adhesion. And it also seemed to prevent alteration of the microbiome, fecal microbiome. So it had, from the literature reports, this compound seemed to have several important properties that might counteract the effects of our microplastic consumption. So to do this, again, the common experimental protocol, we gave mice normal water or polystyrene water. And in this case, we gave injections of either delphinidin or saline over a 4-week time point. And during this time course, we looked at, as our endpoint measures, and during the time course, we looked at glucose levels and body weight. And what we saw as before, as polystyrene increased, polystyrene consumption promoted an increase in weight gain. This was decreased in those mice receiving injections of delphinidin. Similarly, in those mice consuming polystyrene, we saw an increase in percent fat. This is decreased in delphinidin. So this suggests that some common interventions, some antioxidant, anti-inflammatory intervention could counteract the effects of polystyrene consumption. So I think that's all the data I wanted to present, but these studies are just in their infancy, and there are certainly many questions that need to be addressed, many unresolved issues in the field. First of all, as I kind of alluded to in the latter part of my talk, what is the right microplastic type to use? What we are exposed to in nature is not a single plastic type. It is a mixture of types. Secondly, what is the right size and shape? Again, what we see in nature, what we consume, is a whole mixture of different sizes and morphologies. We've used just perfect spherical beads that we obtained commercially. So this is not at all what we're exposed to in nature. Along with the size and shape, what is the right dose? We tried to approximate in our mouse studies the dose of beads that would mimic what's found in a human. Another issue is, again, these beads we get are perfect spheres from a company. This is not what we see in nature. In nature, these plastic beads are oxidized, they're exposed to high temperature, they're exposed to UV, they can carry chemicals and metals, they can carry biofilms. So it's not at all what we see, what we use in animals, not at all what we see in as humans. There's other variables we haven't considered in our exposure system. Are there sex-dependent responses? What are the effects of comorbidities? We did do one exposure with polystyrene beads in mice consuming a high-fat diet. However, we didn't see any difference between in these outcomes. That is, the effects of polystyrene were not worsened or in those mice on a high-fat diet. And finally, are these effects transient or persistent? We need to understand more about the mechanisms. Are these reversible in the absence of exposure with some antioxidant treatment? Are there some epigenetic effects going on which will allow for more persistent outcome? And finally, as I think I've alluded to throughout the talk, is there one single mechanism whereby microplastics can induce their pathological outcomes? Or do they have different mechanisms, impact different parts of physiology? So I think I'll stop here. This work was largely done by Jingjing Zhao, a postdoc in my lab. She had help from my graduate student, Daniel Gomes. There are several people in the Envirome Institute who have helped, especially during the SAC of these animals. Also help from Matt Cave's laboratory, some core facilities here at UofL, the funding from NIH. And there are also several people at UofL who've helped to generate the data or helped me to think about the data and to come up with ideas and plans for future studies. Okay, that's it, and I'll take questions.
Got actually three of them, if you could, they're
Okay.
related. So obviously I'm interested in the cardiometabolic stuff, and the one thing that hit me as you were going through the adiposity, I don't know if you want to bring back, probably don't need to bring back the slide, but the glucose homeostasis actually becomes out of whack in humans, and the insulin levels go up because of the adiposity. So you show that both go up, but the question is do you have an idea which one you think both are being induced by the plastics, or it does one and then the other follows?
Yeah, we don't know that. We haven't done a really good time course to see if one induces the other. From what we've done, it seems like they both come up at the same time. So I don't know if they're independent or of each other or are dependent upon each other. That's something we need to look at further.
So similar to that one, once your insulin level goes up, that's atherogenic, at least in humans. And so wouldn't that explain this? You'd have to have a model where the insulin level was like inhibited somehow to find out if the plastic was really accelerating, or it was just the insulin effect on the blood vessels. Atherosclerosis, insulin levels.
We haven't, like, again, a lot of this probably requires a careful time course study. The only insulin measure we did was at termination. So it may be going up, and it was only in that one group, actually. It may be going up earlier than at termination. And the lesions they may be, we've only done the lesion at termination. We haven't done the time course. So a lot of this, what the sequence events and which promotes which is something we haven't really addressed well.
The last one for me is the interesting slide on decreasing cardiac output. Looked like the ejection fraction went down and the end-diastolic volume went down, which is completely opposite of what happens in humans. I mean, because that really lowers your stroke volume, which is also what you showed. And so the hard part is that if you had a higher adiposity, your cardiac output normally goes up because of that. So this is a striking finding to find. So and the one parameter I was going to suggest is do the cardiac index, and you probably reach statistical significance sooner because it's going in the wrong direction.
That's all we've done in regards to just one echo, and this is something I'm not a cardiologist, so I don't appreciate all these things, but yes, that's
Yeah, well,
an interesting finding. We haven't done, other than this one echo, we haven't really followed up on any cardiac function, but it's, yes, something we should plan to do in the future.
certainly, yeah, it's a scary thing to have your stroke volume go down and diastolic volume go down when your body is going up. Oof.
Yeah.
Okay, all right, fantastic, thank you. Other questions? Go ahead, Dr. Doctor.
So I'm curious, do you think that, like, how far are the microplastics actually getting into us, right? So you get a leaky gut, these the microbiome changes, you get all these kind of physiologic changes, which partly could be the microbiome potentially, some of that signaling, but, I'm thinking about the insulin. How do you like even measure that? Like, are there microplastics getting into the pancreas and that's causing inflammation? Like, what do you think is going on? I guess kind of a bigger global picture.
Yes, I mean, there are ways to measure. We haven't done it. There are ways to measure, as I alluded to in some of the human studies, ways to measure plastic particles in different organs. Paralysis, GCMS. We haven't done that. We don't know where these things go, how much are excreted and how much comes out in the feces. So we don't know where they go. And that's a concern of grant reviewers. But it seems that they go to a lot of places. And how if they have to go to a specific organ to induce pathological outcomes, or whether they can induce oxidative stress in one place and there's some systemic mediator that goes to another tissue is really, not open question. So we don't know where they go in the mice. And again, which pathology comes up first, but it's something we need to address better as to how these particles are distributed. And like I said, what stays, what comes out.
It's kind of striking that your smaller size seems to have
The biggest
some of your
effects.
bigger disruption.
Yeah, and that may be because, it goes to different places that the larger particle doesn't. Yeah.
I'll scoot up a little.
Online?
There's it's more of a comment from Steve Jones. He said, it's hi Tim, interesting and timely work. I, of course, am most interested in the cardiac function data in response to PDS treatment of the drinking water. For the echocardiography data, I consider other interpretations, to wit, rather than indicating hypertrophy. To me, your data indicate potential impaired relaxation. Says I could not see everything in the example of the heart rate, so there could be other explanations. Either way, as always, I'm happy to discuss further with you any cardiac function data.
Okay, thank you, Steve. I'll follow up with you on that.
Great.
That's all.
That's
Okay.
certainly worth looking into, because that is strange findings you just don't see.
Okay, yeah.
Unique
Okay.
data set.
I have a final question.
Yes.
Hi, this has been super interesting. We are working on something really similar in humans, and we're trying to find the relationship between leaky gut microbiome disruption and TMAOs in humans. So this has been like really, really interesting. Talking about the vascular, component of it, I know that, we talked about atherosclerosis and everything. I was just wondering if you have noticed any kind of behavioral or cognitive decline while you were examining this mice, because it has a lot to do, right? And we also have found some data about butyric acid and the cognitive part. So I was just wondering if you found or if you're planning to do something with that kind of data
Yeah, we haven't done any kind of behavioral studies,
or
but
cognitive.
no. And although that would be interesting in light of this, as I mentioned, this Nature Medicine paper where they found high levels in the brain. So
Yes.
interesting, yeah.
Because I mean, that might be something.
Which brings me to maybe a final point. As mentioned, I'm focused mostly on the vascular outcomes, but we collect a lot of organs, and anybody out there wants to have their own favorite organ system or pathology, we're happy to collaborate, so. Okay.
Thank you very much.
Sure.
Yeah, okay. So,